everyone, and welcome to the third lesson in National 5 Biology. We're continuing on with Key Area 1 of the first unit, which is cell structure, and today we're going to be looking at microscopes and stains. So to begin with, just remembering that the icon in the top left corner, the pencil and pad, that basically means that this is something that I would want you to remember. So maybe take a note of this slide. Stains are used to show the detail of a cell under the microscope. So in the picture on the right hand side, these are cells looked at underneath a microscope, and you can see they have the nucleus, you can see their cytoplasm, and you can see that they have a distinctive edge surrounding the cell, and that would be the cell membrane. These are animal cells and they have been stained blue. They are blue in colour now because of the methylene blue stain. And plant cells we stain with iodine, which is orange in colour. So it's iodine used to stain plant cells and methylene blue used to stain animal cells. Now we have a diagram of a microscope. So the different parts of the microscope are, starting with A, we have the eyepiece. And inside of the eyepiece there is a lens. So we have the eyepiece lens. B and C are both used to focus in on the image that you're looking at. So they'll make them go from a blurry image to a nice clear image where you can see lots of detail. B is the larger wheel, so it is the coarse focus. And then C is the smaller wheel, which is the fine focus. So the coarse focus would be used to make large movements. So you'd use your coarse focus to first find the cells that you're trying to look for. Before that, it would maybe just be like a clear screen that you're looking at. You then use your coarse focus to actually find the slide that has the cells on it. And then you would use C to fine-tune that image to take it from a blurry image to one that's really focused and detailed, where you can see all of the different details and it's really sharp and clear. D is our objective lens. So you have a lens up top in A, so that's your first lens. And then you have a second lens. So when you're looking down a microscope, typically everything is magnified twice. So first with the eyepiece lens and then second with the objective lens. And usually you can change your objective lens. So there might be 1, 2 or 3 different options for your objective lens. And that would usually allow you to move up magnifications to look at your slide in a greater magnification. You'd start off at the lowest and then move until you reach the highest. E is where you sit your slide, it's where everything happens, and that is the stage. Okay, if you were going to see like a theatre show, you would look at the stage. Just like in the microscope, you sit your slide on the stage and that is what you look at, that's where all the action happens. And then F can change depending on what type of microscope you're using. So this one has a mirror. Other types of microscopes might have a light bulb down the bottom. So basically all F is a light source. So our eyepiece lens and our objective lens, 2 different lenses in the microscope. You have your 2 focus wheels, the coarse and the fine focus. You have the stage where you sit your slide, and then a light source which shines light up through the slide, through the lenses and into the eyepiece so that you're able to see what you're looking at and it's not just dark. So a microscope is used to make small objects appear bigger than they are. It magnifies what you're looking at. And in order to calculate the total magnification of a microscope, you times the eyepiece lens with the objective lens. So an example of that would be this one here where the eyepiece has a magnification of 10 times. So times here, this X, is a unit. Okay, so just like if you were doing length, you could have maybe 40 centimetres or 400 millimetres. They would be an example of units. The unit for magnification is times. So 10 times something, 40 times something. And the way you would write that is you'd add the X next to the number. So we need to remember our units when we are giving answers. So the eyepiece has a magnification of 10 times and the objective lens is 40 times. So to calculate the total magnification, you would times the eyepiece by the objective, and that would be 10 times by 40, which will give you 400 times total magnification. One more example, we have the eyepiece is a magnification of 5 and the total magnification is 50. It now wants you to calculate the magnification of the objective lens. So you have your eyepiece times objective equals the total magnification. So we know that it's 5 times something equals 50. So all we need to do to find out what this something is, what our objective lens is divide by 5. And that gives us 10. So that means that our eyepiece is 5 times by 10 to equal 50. Okay, so that one is working backwards. And then to summarise everything, stains are used to show the detail of cells under the microscope. We use iodine for plants and methylene blue for animals. And then to calculate the total magnification of a microscope, you use the eyepiece times the objective lens. Okay, and sometimes you may be asked to work backwards. So you need to remember, if you're given the total magnification, then you're dividing instead of multiplying. Okay everyone, I hope that this lesson has helped. I hope that whether you are learning or revising, this has been useful to you. This is the end of cell structure. The next lesson will be on key area 2 of Unit 1, which is the transport across cell membrane, so stay tuned for that. Thank you guys all for watching, and I will see you in the next lesson. Bye everyone.